Hello, good evening, students. How are you? Hope all of you are doing well. So today is our second class, and we have to discuss about that is the functional and structural unit of life, that is cell. So please make sure that audio and video is clear, and let me know first. So shall we start? So we have to discuss about what is cell, and about the history of cell, how they get discovered, and what are the function of cells, what are the organelles present inside the cells, and what are the function of different organelles present inside the cell. Plus we have to discuss different types of cells, we have to discuss the difference between plant and animal cells, and we have to discuss the basically the content present in the cell, mainly nucleus. Fine. So first of all, introduction. So cell is the smallest unit of living things. Now if we talk about the science, we call smallest unit as atom, but atoms are the things which basically comprise to make a matter. If we talk about the living organism, the smallest unit in the living organisms are cell. These cells are the structural, means they are forming the structure or the whole body of a living organism, and also functional unit of living organism. Plus they are performing some kind of function due to which a living can sustain their life. Plus these cells are the fundamental, or we can say that is basic unit of living. Any living organism in this world is basically made up of what? Cell. So it is the smallest unit. Fine. Now if we talk about the history of the cell, there was a scientist in 1665. His name was Robert Hooke. What he have done, he was working over some plant. So what he have done, he have taken the cork, which are the dead cells of the plant, or usually these corks are put it over the bottle of the champagne. You have seen the cap over the bottle of the champagne. So what he have done, he cut it fine slice of those cork cells, and he observed those structure inside his own made microscope. So let us talk about the history or discovery of cell. So in the year of 1665, a scientist Robert Brown, what he have done, he cutted down fine slice of cork, and cork are the dead cells or the dead structure which helps in the formation of the bark of a tree. Fine. So what he have done, he cut it down the fine slice of cork, that is dead cellular structure, helps in forming bark. So all of you have seen a tree. Or at the upper surface of the tree, you have found a hard sclerite structure or a hard structure which is commonly known as bark of a tree, जिसे हिंदी में छाल बोलते हैं. So he cut it down the fine slice of cork and observed these slices in his own made simple microscope. Now, what are simple microscope? If a microscope contain only one set of lens, only one type of lens, such type of microscope are said to be as simple microscope. So in the year of 1665, Robert Brown cut it down fine slice of cork. What are cork? These are the dead cell structure which helps in forming the bark of a tree, and he observed these slices in his own made microscope. What he have observed? He observed little compartments looking like honeycomb. So under his observation, he found some little compartments or room which are looking like a honeycomb. So in Latin language, these honeycombs, in Latin language, these honeycomb like structure are said to be as cella. So the word cell derived from which language? Latin language. And in English we call it as cell, which is the structural and functional unit of life. Is that clear? Is that clear? So again, let's start the introduction of the cell. What is cell? It is the smallest unit of living things. These are the structural and functional unit of living things. Means a living organism, okay, is made up of cell, and something is working inside these living organism, and these structure we commonly call them as cell, and these cells are the fundamental or basic unit of living organism. If we talk about the history or discovery of the cell, in the year of 1665, a scientist named as Robert Brown, he cut it down the fine slice of cork, and these corks are basically dead cell structure which helps in forming the bark of a tree. He observed these slices in his own made simple microscope. What are simple microscope? If a microscope made up of only one type of lens. Nowadays, microscope have more than two type of lens. So earlier, the primitive microscope have only one type of lens. So such type of microscope which have only one type of lens are said to be as simple microscope. These microscope are used to basically observe the dissected structures of plants and animal. Dissected means cutted structure or the cutted part. So under his observation, he found little compartment, and these little compartment looking like honeycomb. तो मधुमक्खी के छत्ते जैसे दिख रहे थे. So in Latin language, these honeycomb like structure are known as cella, and in English we convert this cella into a simple terminology, that is the cell, which is the basic or the fundamental unit of life. Is that clear to everyone? Is that clear to everyone? So, Robert Brown is said to be as the father of cytology. What is cytology? Study of cell. Cyton means cell, logos means study. The study of cell and its internal environment or cellular organelles is said to be as cytology. Now, but the problem with, oh sorry, not Robert Brown. I'm extremely sorry, Robert Hooke. Robert Brown have discovered the nucleus. Extremely sorry. Robert Hooke, not Robert Brown. So again, history. In the year of 1665, Robert Hooke cutted down the fine slice of cork, which are the dead cell structure, and these dead cell structure helps in forming the bark of the tree. And he observed these slices in his own made simple microscope, and these microscope contain only one type of cell. What he have observed, little compartment or little room like structure or honeycomb like structure. And in Latin language, honeycomb like structure are termed as cella, and from cella we have converted it into an English word terminology, that is cell, which is the basic unit of life. So Robert Hooke is said to be as the father of cytology. Next thing is, but the problem with Robert Hooke discovery was that Robert Hooke has observed dead cell. As we have already discussed that what he have done, he cut it down the fine slice of cork, and these corks are the dead cellular structure. Now, so Robert Hooke has observed the dead cell. So he was the father, no doubt he was the father of cytology, but he did not observe the living cell. So the credit for observing the living cell for the first time goes to a different scientist. Now, in our exam, usually a question asked, he published the image of first cell in a book known as Micrographia. So in our exam, usually they ask a question, in which book Robert Hooke has published the image of first cell, whether it is Historia Animalium, whether it is Systema Naturae, whether it is Origin of Species, whether it is Micrographia. So the answer to this question will be Micrographia. So let me show you the image of the first cell. These are the images of the first cells which was published under the book known as Micrographia, and these images are giving the appearance of a honeycomb like structure, the hive which are formed by the honeybees, or the home which are made by the honeybees. So the credit for observing first living cell goes to a different scientist known as Antonie van Leeuwenhoek. So Antonie van Leeuwenhoek, he was a worker in a glass factory. What he have done, he have designed a new type of microscope which contain more than one type of lens, and he started observing uh, the pond water, the blood, the semen, then the drainage water, and he discovered various different types of cell. So the credit for observing first living cell goes to Antonie van Leeuwenhoek in the year of 1674. So first living cell was discovered in the year of 1674. Fine. Now, the first living cell observed by Leeuwenhoek was pond algae, which is commonly known as Pyrogyra or pond silk. Now, what he have done, he first of all collected the sample of water from a pond nearby to his factory, and he started observing the greenish growth which is present over the pond. These growths are basically small microscopic structure which perform the or which have the ability of photosynthesis. These structure or these organisms are known as algae, and the name of pond algae is Spirogyra. The algae which we see usually over the pond, its name is Spirogyra, and it is commonly known as pond silk too. So if somebody is ask you which algae is known as which algae is known as pond silk, now they'll give you four different options like Volvox, Chlamydomonas. These are the name of certain different algae. Spirulina and Spirogyra. So the correct answer to this question will be, that is, the pond algae is Spirogyra, and these Spirogyra are the first living cell observed by Antonie van Leeuwenhoek in the year of 1674 by his self-designed compound microscope. So the idea of using more than one lens in a microscope was also given by Antonie van Leeuwenhoek, and this guy, that is Antonie van Leeuwenhoek, also known as the father of microscopy. And the microscope which we usually find in our school labs or normally in pathological labs are generally compound microscope which contain more than one type of lens. Is that clear? Is that clear? Is that clear to everyone? No. Next thing is A.V. Leeuwenhoek also discovered RBC, that is red blood cell from our blood, semen, oh sorry, not semen, sperm from semen, and bacterium or bacteria from drainage water. But he coined the term small animalcules. What he have done, he started observing the drainage water, नाली का पानी देखना चालू किया, and he have observed that something is moving inside that water which is not visible to the naked eye but can be seen through that microscope designed by Antonie van Leeuwenhoek. So he called them as small means very extreme fine particle animalcule, animal like substances, because only at that time we just know only animals can move. Later on we have discovered many things which can show the property of locomotion and movement. So small animalcules. So earlier bacteria was termed as small animalcule. Later on another scientist, Ehrenberg, coined the term bacteria in place of small animalcule. So if somebody is asked you who coined the term small animalcules for bacteria, so small animalcules term for bacteria was coined by Antonie van Leeuwenhoek, but the term bacteria was coined by Ehrenberg. Is that clear? Is that clear? Next thing is now various events occur. Grew and Malphizi were the another scientist who was first of all observed the plant cell. Then various different scientists came and give different kinds of theories related to cell. Those all theories are not present in your syllabus or are basically they are not for the competitive exams. We look over what is mentioned in our competitive books or the NCRT. Then two different scientists in the year of 1839 proposed a theory. This theory is known as soil theory in the year of 1839 by two different scientists, by Schleiden and Schwann. Schleiden was a botanist, means he used to study the plant. Schwann was a zoologist, means he used to study the animal. So what happened in the year of 1838? Schleiden proposed a statement that is all plants are made up of cell. As plants are the living things, so they are made up of cell. In 1839, Schwann gives a statement all animals are made up of cell. In 1839 only, both Schleiden and Schwann proposed a new statement in biological world, and this statement is all living things are made up of cell, because plants and animals both are the living things. So in 1839 they modified these two statements into a one statement, and they said all living things are made up of cell, and this statement is said to be as the cell theory. Is that clear? Is that clear to everyone? Fine. Now, there are certain postulate means points which comes under the cell theory. Number one is all living things are made up of cell. All living things are made up of cell. Number two, cell is basic या फिर fundamental unit of life. We have already discussed this point. Number three, cell is structural and functional unit of life. Number four, we have already discussed these three different points. Number four, cell show different metabolic reaction. So inside cell there are different types of reaction occurring, like there is synthesis of protein, there are synthesis of various cell organelle, there are transportation of protein, there are signaling, there are apoptosis, there are translation, there are replication, there are n number of activities going on inside the cell, and these all activities come mildly known as metabolic activities. So cell shows different metabolic activities. So these are the certain point or the postulates in cell theory. Now, all living organism must obey this theory, which is commonly known as cell theory. Means we all are living organism, we are obeying what? Cell theory. Plants are living organism, they are also obeying cell theory. Fungus are living organism, they are also obeying cell theory. Protista means amoeba, parameciums are living organism, they are also following cell theory. Even bacteria are the living organism which are following the cell theory. But there are certain exception to cell theory. Means these are those organisms which do not follow cell theory, and these organisms are commonly called as viruses, because cell theory must be or should be obeyed by only living organism, and viruses are neither living nor non-living type of organism. What are viruses? These are link between living and non-living. Outside the cell or outside the host body, viruses are non-living thing, and inside the host body or inside the cell, viruses are living thing. So these are the exception to the cell theory. Viruses never follow cell theories, they are not cell. So you will not find any kind of cell organ like mitochondria, ribosome, then plasma membrane, then Golgi bodies. These type of cellular organelles are not seen inside the viruses. Is that clear? Is that clear? Now, next thing. In the year of 1855, another scientist, Rudolf Virchow, proposed modern cell theory. So what he have done? He have done nothing. He point out all the points of cell theory inside modern cell theory. He just added one new point inside the cell theory, and he wrote that point in Spanish language like this: Omnis cellula e cellulae. This is Spanish. And if we convert this Spanish into English language, this is what? Spanish. This is the new point of cell theory. If we convert it in English, the means of omnis cellula e cellulae is, that is, new cell arises from pre-existing cell. So whenever, let us suppose this is the older cell, whenever it is about to die or it is about to complete its cycle, or it get ruptured by some kind of shock, like by accident, or some acid is fallen over it, these cells started division or proliferation and start forming different type of start forming the daughter cell. These are the daughter cell and these are mother cell. So this idea of cell division is the basically new pointed added inside this old cell theory with a statement that is omnis cellula e cellulae, and the meaning of omnis cellula e cellulae is, that is, new cell arises from pre-existing cell. Is that clear? Is that clear? So this is modern cell theory, omnis cellula e cellulae, which means new cell arises from pre-existing cell. Next thing is, now let's talk about shape of cell. The shape of a cell is not fixed. These shapes are decided on the basis of their function. A cell can be oval in shape, or not, sorry, oval, spherical in shape, oval in shape, comma like, star like, wavy, spiral like, irregular, so a cell basically do not have any fixed type of shape. It can be of n number of shape. We have polygonal type of cell, we have spherical type of cell, we have oval type, we have comma like, we have star like, we have spiral like. So there are n number of shapes for the cell. Fine. Now let's talk about the number of cell. Depending upon the number, a living organism can be unicellular, means made up of only single cell, or multicellular, means made up of more than one cell. For a kind of example, bacteria are unicellular organism. Protozoa means amoeba paramecium like organism are unicellular organism. But if we talk about plants, plants are multicellular organism. Animals are multicellular organism. Fungi are multicellular organism, except there are one exception in kingdom fungi, that is yeast. The yeast which is used in our home to make cake, to make jalebis, to make dosa, or also used in bakery industries as well as in alcohol industries. These yeast are unicellular organism. All fungus are multicellular except yeast. Is that clear? Is that clear to everyone? So we have shape of the cell. The shape of the cell is not fixed. It depends upon its function. In our living world, we have spherical, oval, comma-like, star shape, octahedral, polygonal, icosahedral, spiral, irregular shape. Different types of shapes are available in our living world. If we talk about the number of cell, according to the number of cells present in the organism or present in a living organism, there are two different types of living organism. They can be either unicellular, made up of single cell, or they can be either multicellular, made up of more than one cell. Bacteria, protozoa means amoeba paramecium like organism, are is unicellular organism, while plants, animal and fungi except yeast are multicellular organism. Is that clear? Is that clear to everyone? No. Next thing is the type of cells. Now, in our living world, there are generally two types of cell. Although in higher studies there are three types of cells, but in NCERT only two types of cells are mentioned. That's why I'm gonna tell you only two types of cell. One is prokaryotic cell, another is eukaryotic cells. These prokaryotic and eukaryotic are again derived from Greek languages, in which pro means primitive and karyon means nucleus. Means those cells which are having primitive nucleus without boundaries are said to be as prokaryotic cells. While in Greek word eu means true and karyon means again nucleus. Means those cells which have well defined nucleus surrounded by boundaries or membranes are said to be as eukaryotic cells. This term was given by, means these prokaryotic and eukaryotic terms was given by Cheton. So these prokaryotic and eukaryotic term was given by another scientist known as Cheton. Now, what do we mean by a prokaryotic cells? Means which cells comes under, or which organism comes under prokaryotic cell and which organism comes under the eukaryotic cell. So let us study the difference between a prokaryotic and eukaryotic cell. So we are gonna classify, or we are gonna make some points, and on the basis of those parameter or points we describe both prokaryotic cells and eukaryotic cells. So at one side of the board I am writing about prokaryotic cells, while on the other side I am writing about eukaryotic cell. Now, the points under which we are going to classify these two different types of cell, or the parameter under which we are going to classify these two different type of cells. First point is size. The size of prokaryotic, because these are the primitive types of cells, or these are those cells which come first during the evolution of life or during the origin of life. So the size was very small. So it ranges from 0.1 micron to 10 micron. Micron means 10 to the power minus 6. While eukaryotic, 10 to the power minus 3. While eukaryotic cell ranges from 10 to the power, not 10 to the power, 5 micron to 100 micron. That means the size of eukaryotic cells are large comparison to the prokaryotic cells. So this is the first parameter on which we have classified prokaryotic as well as eukaryotic cells. Next thing is size, then nucleus. In prokaryotic cells, nucleus is absent, but we find a nucleus like structure, and these nucleus are like structure are said to be as known as nucleoid. Now, in any biological term, if you find oid at the suffix or at the suffix position or at the end of the word, it means like that. Like that means like that. Like that, the word which is written before it. Like cuboid, like cube, cube like structure. Rhomboid, rhombus like structure. Spherozoid, sphere like structure. Nucleoid, nucleus like structure. So actual nucleus is absent, but we find a nucleus like structure which works as a nucleus, and these structures are said to be as nucleoid. Here we found true nucleus surrounded by membrane, and due to the presence of these membrane, what are these membrane? Nuclear membrane, which is enclosing the nuclear material, we can tell that this part is nucleus and it is easily visible. But in prokaryotic cell, we do not find these nuclear membrane, due to which nucleus we cannot distinguish between what is nucleus. That's why there is a structure without nuclear membrane, and we call that structure as nucleoid. Next thing is membrane bound organelle. We are talking about cell organelle, those organelle or those structure which we found inside the cell, like mitochondria, like endoplasmic reticulum, like Golgi bodies. These are bounded by membrane. So in prokaryotic cell, these structure are absent. There is no mitochondria, there is no Golgi body, there is no endoplasmic reticulum, there will be no what क्या बोलते हैं उसको, lysosome and other structure. There will be no vacuoles. So that means these structure are absent in prokaryotic cell, but these cellular organelles with a proper membrane bound structure is present inside the eukaryotic cell. Next thing is ribosome. Now ribosome are those cellular organelle which helps in the formation of protein. In prokaryotic cells, we have 70 S type of ribosome. This S is nothing, it's a symbol given by a scientist known as Swedberg, and these are sedimentation rate of these structure under a device, and that device is known as centrifuge. You have usually seen these devices in dairy. आप सभी ने देखा होगा डेयरी में एक चक्का लगा रहता है जिसमें दूध के सैंपल घुमाए जाते हैं, ठीक है, that was centrifuge. So under that centrifuge, at what rate these particles sediment inside those vials or tube, that's why we have given this symbol S, sedimentation rate, yeah, or Swedberg constant. And in eukaryotic cells it is 80S. But these structure 70S and 80S only form during the time of protein synthesis, when ribosome have to synthesize a protein. Otherwise these structures, these ribosomes remain apart in the form of large unit and in the form of small unit inside a cell. Also here large unit means until or unless a cell is not forming a protein, these ribosome remains apart in the form of large or small unit. As soon as they have to synthesize the protein, these large unit and small unit combine to form 80s ribosome in eukaryotic cells and 70s ribosome in prokaryotic cell. So the large unit in prokaryotic cells is 50s, while the smaller unit in prokaryotic cell is 30s, and the larger unit in eukaryotic cell is 60s, while the smaller unit is 40s. Don't get confused and don't use your mathematics. If we add 50 and 30, there will be 80, and sir is writing 70s over here. If we adding 60 and 40, there will be, the product is 100 and sir is writing 80s over there. It is like this only. These are conformational changes, a 3D thing which I cannot show you over the board. You just have to memorize. During the time of protein synthesis, prokaryotic cells have 70s ribosome, which are formed by the aggregation or combining larger unit 50s and smaller 30s, and in eukaryotic cell these are 80s, which are formed by large unit 60s and small unit 40s. And the example of these cells. Now, what type of organism are comes under the prokaryotic cell, or we can say that which living organism have these types of cell? So all bacteria of this nature are prokaryotic cell, and these bacteria are the most primitive organism present in our earth, which forms during the evolution or origin of life. While protozoa like amoeba, paramecium, fungus, plants and animals, these are eukaryotic cells, means their size is around 5 micrometer to 100 micrometer. They have true nucleus surrounded by nuclear membrane. They have membrane bound organelle. They have 80s ribosome. All these organism shows these common feature. But bacteria shows the size should be around 0.1 micron to 10 micron. They don't have nucleus, they have nucleoid in place of nucleus. They don't have membrane bound organelle. They don't have mitochondria, Golgi body. Ribosome is 70s type, and the example of prokaryotic cell is bacteria. Last thing of today's class, or last thing of the introduction to cell, that is largest cell. The largest cell of this living world is ostrich egg, while the longest cell of this living world is found inside our body. These are the cells of our nervous system, and these cells are known as neuron or nerve fiber. And the smallest cell in this living world is bacteria, whose size is 0.1 micron. This is mycoplasma. This is the name of the bacteria, Gallisepticum. Or also known as PPLO, that is pleuropneumoniae like organism. So this is all about the introduction to cell. In our next turn, that is will be on Monday, we are going to discuss about the different cell organelles found inside the cell, like chloroplast, mitochondria, plasma membrane, vacuoles and other different structure. So I hope you have understand well about the introduction of cell. Now let's meet on Monday. So till then, have a nice day. Bye bye. Good day. Take care. Thank you.